Hello, welcome back to my channel, Bio with Shansi. I'm glad that you are here. Just know you have made the right decision. In today's lesson, I am going to cover, or we are going to look at what we call globular proteins. We're going to look at examples of globular proteins and fibrous proteins. So let's dive right into it. Globular proteins, globular. Globular is coming from the word, you could think of a globe. So when you're told to describe a globular protein, we are looking at a protein that is like a globe, a ball-shaped protein, or a protein that is spherical in nature. This means that the polypeptide chains of that protein are curled up into a ball-like shape. And examples of globular proteins, we have hemoglobin and myoglobin. Now, when you are told to describe a globular protein, remember it is spherical in shape, or you can simply say it is a protein whose polypeptides curl up into a ball-like shape or a spherical shape. So go ahead and mention when these polypeptide chains are curling up, how do they curl up? So the curling up happens in a way that you are going to find that when you look at the amino acids that make up the polypeptides, the amino acids are arranged in a way that the hydrophilic amino acids are going to be on the outside. Why is it so? These globular proteins most of the time are going to be found in watery environments. An example, hemoglobin is found in blood. So it should be soluble because blood is made up of water. You'll find some of the proteins found in maybe the phloem, that is in plants or in tissue fluid. So these proteins have to be soluble. And what is going to make them soluble are the hydrophilic amino acids. So these hydrophilic amino acids, like in this picture, the blue ones are the hydrophilic amino acids. They are going to be surrounded by the watery environment. So they have to be really soluble. So the amino acids in a globular protein are arranged in a way that the hydrophilic amino acids are going to be found on the outside, whereas the hydrophobic ones are on the inside. So the hydrophilic ones will make the molecule soluble. Why? Because the water molecules will get attracted to the hydrophilic amino acids. But the hydrophobic ones, remember, are insoluble, so they tend to hide away from the watery environment. And this means that water molecules can never reach the center of this protein. So the water molecules are going to keep being repelled away from this center, and they will only remain on the outside. So that is how a globular protein is. These proteins curl up so that their nonpolar hydrophobic arrow groups point in the center away from the watery environment. And the hydrophilic amino acids or the hydrophilic arrow groups are going to be found on the outside. So the fact that we have the hydrophilic amino acids on the outside makes these proteins much more soluble, or they are going to be soluble, since most of the time they are found in watery environments. So these globular proteins have different they have different roles that they play in chemical reactions, and their shape is very key in the functioning. Remember, the shape of these globular proteins is still held by the same bonds, the hydrogen bonds, the disulfide bonds. Now, an example here in the center, we shall have hydrophobic interactions between the hydrophobic amino acids and the ionic bonds. So enzymes are examples of globular proteins. Hemoglobin is an example of a globular protein, and we are expected to know one example of a globular protein, which is basically hemoglobin. So hemoglobin is a globular protein. Still, if I'm asked to describe hemoglobin as a globular protein, I'll simply say hemoglobin is made up of polypeptide chains that are curled to form a ball shape or a spherical shape. Then go ahead and describe how does the curling occur. The curling occurs in a way that the hydrophilic amino acids are on the outside of the molecule, whereas the hydrophobic ones are on the inside of the molecule. Then mention that hemoglobin is going to be soluble. So hemoglobin is made up of a globular structure. So it is a globular protein. Now, hemoglobin is, I'm sure you know that it's found in red blood cells, so its role is to carry oxygen. This hemoglobin molecule is made up of 4 polypeptide chains. So it shows a quaternary structure. We saw that in our previous lesson. It shows a quaternary structure because it is made up of 1, 2, 3, and 4 polypeptide chains. And then it also has a non-protein group, which is called a prosthetic group. So hemoglobin is made up of a non-protein group, and the non-protein group still makes it qualify to have a quaternary structure. So hemoglobin is called heme. When you look at the word heme, heme comes from the iron, which is attached to it. Each polypeptide has an iron atom attached to it. So that is why it is called heme. Then globin comes from the fact that the polypeptide chains are made up of a protein called globin. So it has 4 polypeptide chains. 2 of the chains are made from alpha globin, and then 2 of the other chains are made from beta globin. Please do not confuse beta globin with beta pleated sheets and alpha globin with alpha helices. No, they are totally different. So alpha globin is a special type of protein, and even beta globin is also a type of protein. So those 2 proteins are the ones that make up hemoglobin. So hemoglobin has two polypeptide chains made from alpha globin, and the other 2 polypeptide chains made from beta globin. The molecule is spherical, meaning it's a globular protein, and the 4 polypeptides pack close together. They are organized in a way that the hydrophilic amino acids will be on the outside. So on this outside, we shall have hydrophilic amino acids, and in the center, we shall have their hydrophobic amino acids. So this hemoglobin molecule, we have seen each polypeptide has an iron group or a heme group. And the role of hemoglobin is to carry oxygen. So when oxygen is to be carried, it is carried by the heme group. So each heme group combines with 1 molecule of oxygen. That means that 1 hemoglobin molecule is able to carry 4 oxygen molecules. 4 oxygen molecules or 8 oxygen atoms. Remember, heme is non-protein, so such a group as heme, which is part of a protein, is called a prosthetic group. And such a group is very important for the protein to perform its role, like the iron is very important for hemoglobin to carry oxygen. So each heme group carries 1 oxygen molecule. So you find that a red blood cell could have like millions of hemoglobin molecules in it. And as a result, it will be able to carry as much oxygen as possible. So oxygen usually binds to the heme group to form oxyhemoglobin. Hemoglobin that has combined with oxygen is called oxyhemoglobin. So these polypeptides, the polypeptides are made up of amino acids, and we have seen that the amino acids on the outside have to be hydrophilic, and the ones in the center have to be hydrophobic. Now, remember we saw that the primary structure of a protein is very important in determining its shape and then how it's going to function, and it determines how the protein is going to be folded. So here they are giving us an example of a scenario where we have the primary structure in hemoglobin slightly changing by just one amino acid, and that brings about sickle cell anemia. So here they are telling us that one amino acid which occurs on the surface of one of the beta chains is replaced with a different amino acid. Remember, on the surface, you have to be there with hydrophilic amino acids. But in this case, in sickle cell anemia, people who have sickle cell anemia, for them, instead of having a hydrophilic amino acid on one of the beta chains, they have a hydrophobic ones because of mutations. So the right amino acid is glutamic acid, which is polar or hydrophilic, but it was replaced by valine, which is non-polar. And now having a non-polar amino acid on the outside of the molecule makes the molecule much less soluble. And that eventually even alters the shape of the red blood cell that is having such an amino acid in its hemoglobin. So that brings about the sickle cell anemia and those associated dangerous symptoms that comes with sickle cell anemia. So this shows you how much important the primary structure of a protein is in determining the function, actually determining the shape first and then the function of that protein. So that is hemoglobin as an example of a globular protein. We also have what we call fibrous proteins. Fibrous, from the word fiber. So these are proteins that just form strands, like fibers. They do not curl up into a ball, but for them, they just form long strands. Let's look at a very common example that we all have, hair. You can see that hair is fibrous. It has those long strands. So hair is a fibrous protein, it's keratin. And fibrous proteins, most of the time, are insoluble. They do not dissolve in water. I'm sure you always go and shower, but you have never seen your hair dissolving. Why? Because it's a fibrous protein. That means that for them, they are made up of non-polar amino acids or amino acids with non-polar aryl groups. And these fibrous proteins, most of the time, provide a structural role. They provide protection, they provide structural support to the different parts of the body where they are found. Your hair is fibrous. Your nails are fibrous. When you look at your skin is even waterproof. So it's also made up of keratin, which is a fibrous protein. And one example of a fibrous protein we are interested in learning about, or the syllabus requires you to know about, is collagen. So let's look at collagen as an example of a fibrous protein. When you look at collagen, collagen is very abundant in the different parts of our bodies. Like you find it in the hooves of animals. You're going to find it in our veins, in our arteries, in tendons, in bones, in shells of different organisms. So collagen is very important as a structural protein, providing support and protection to the different parts of the body where it is found. So this collagen is insoluble. Look at your skin. Your skin never dissolves. When you go into a swimming pool, I'm sure you have never seen yourself dissolving because your skin is waterproof due to the collagen there. Leather is collagen. Cartilage, bones, your teeth, all those are made up of collagen. So it is a very important structural protein in animals, and it can be found in different parts of the body. So its major role is to provide a structural function. So you're expected to describe the structure of a collagen molecule. That takes us to the next slide. Now, a collagen molecule, first things first, is made up of 3 polypeptides. 1, 2, 3. You can see here we have 3 polypeptides. So a collagen molecule is made up of 3 polypeptides. Now this shows that collagen also shows a quaternary structure because it has 3 polypeptide chains. Now when you look at these polypeptides, each one of them coils to form a helical kind of structure. So that is why you find collagen being described as having a triple helix. Triple because it is made up of 3 polypeptide chains and they all coil around each other so tightly, forming a three-stranded rope. Three-stranded rope. And that three-stranded rope is what we are going to call a triple helix. A triple helix. Now, when you dive into the amino acids that form each polypeptide chain, you'll find that the amino acids, each third amino acid is glycine. I don't know whether I remember about glycine, that we saw that glycine is one of the simplest amino acids with just a hydrogen as an arar group. It's very simple, with only a hydrogen as an arar group. Remember that other amino acids have very much bigger arar groups, but glycine is the simplest with hydrogen. Hydrogen is a small molecule. So each third amino acid is glycine. That is one of the other properties of collagen, that when you look at the polypeptide chains, each third amino acid is glycine. So these 3 polypeptide chains are coiled around each other, forming a very tight coil. And that is why collagen is said to have a triple helix. So those coils are held tightly together by hydrogen bonds and some covalent bonds. Yes, they are held together by hydrogen bonds. So why is it important that each third amino acid is glycine? Look at these coils. They are so tight. And the fact that each third amino acid is glycine, which is the simplest amino acid, means that these 3 polypeptides can tightly be wound around each other compared to if there was a bigger amino acid. If there was an amino acid with a bigger arar group, the tight coiling couldn't be possible. Think of this: when you are hugging someone who is small, you can easily give them a tight hug because their body is small and you can easily wrap yourself around it, right? But imagine you're hugging a pregnant woman. There is no way you can hug a pregnant woman because obviously their belly is going to push you further away. So you cannot form a tight hug with someone who is pregnant. So this is the same case for collagen. These collagen molecules, the 3 polypeptide chains, are able to be tightly wound around each other because each third amino acid is glycine, which has a small arar group. So that small arar group allows that tight coiling to occur between the 3 polypeptides. So that is it about a collagen molecule. Remember that a collagen molecule has 3 polypeptide chains. Each chain is in shape of a helix. We have seen that 3 polypeptide chains are wound around each other, forming a triple helix, which is like a 3-stranded rope. At the same time, the 3 strands are held together by hydrogen bonds and covalent bonds. And we have also seen that almost every third amino acid is glycine, which is the smallest amino acid. So this small amino acid allows the 3 strands to be so close together so that they can form a tight coil, because any other amino acid would be too large and wouldn't allow that tight coiling to occur between the 3 polypeptide chains. So that is a collagen molecule. Now, usually we can have these collagen molecules. Remember, this is one collagen molecule, another collagen molecule, and another collagen molecule. They can lie parallel to each other and form what we call microfibrils or collagen fibrils. So here we are having one collagen molecule lying parallel to another collagen molecule, lying parallel to another collagen molecule. So many of these triple helices are going to lie side by side, and we can see they have covalent bonds linking them. They have covalent crosslinks that are holding them together. You can see this is held to the other by these covalent bonds. So we have those covalent bonds holding them. So these covalent bonds are very much important in making sure that the collagen molecule is strong, or in making sure that whichever structure contains collagen is going to be provided with the strength that it needs. So from this, we have seen that each three complete three-stranded molecules of collagen interact with other collagen molecules running parallel to it. And collectively, they form what we call fibrils. So in these fibrils, we have seen that the collagen molecules are connected to the ones lying parallel to them by covalent bonds. But let's notice one thing about these covalent bonds. The covalent bonds are all not at the same level. You can see the covalent bond. We have this one right here. Another one is in a different position. Another one is in a different position. And even when you look at the gaps between the collagen molecules, the gaps are all not at the same level. So we can say that the ends of the molecules are staggered. Staggered means that they are not at the same level, they are at different points. You can see this end is here, then the end for these two is here, the end for this is here. So that staggering allows this collagen molecule not to have any weak spot running right across the fibril where the collagen molecules are found. So this makes collagen much more strong, because if all these ends were on the same level, that means there would be a weak point where the collagen molecule could easily break. But usually, collagen is very strong simply because the ends of the molecules are staggered. They are all not at the same level. They are all not at the same level. So many collagen molecules form fibrils. So this is a fibril. Now we can have many of those fibrils lying alongside each other to form now fibers. To form fibers. So collagen has a structural role. It is flexible, however, it has a very high tensile strength, meaning that it can withstand pulling. Try to pull your bones and find out whether your bones are going to break. They will not break. They will not stretch. Why? Simply because they are made up of collagen. Here they are giving us an example of the human Achilles tendon, which is found in your legs. So this tendon is very strong because it has a lot of collagen in it. And they are equating its strength to steel. You can imagine it being as strong as steel. This means it can withstand the pressures, because usually you're moving, or you could be running, exercising, and it is going to be providing support and strength to your legs. And these collagen fibers will line up in the body depending on the forces they are expected to withstand. Like in a tendon like this, they're going to be lying parallel to each other. In the skin, they're going to be running in different directions, because your skin is exposed to different forces that are coming in different directions. So if those are the collagen fibers, they are going to be in the skin, they are going to be lying in different directions so that your skin is able to withstand pressure or forces from different directions. Forces from different directions. So this collagen is very important as a fibrous protein. You should be able to describe the structure of collagen. So here we are having a scanning electron micrograph of collagen fibrils. So these are fibrils, one fibril, another one, and this banded appearance shows you the staggered gaps between the collagen fibers. So that means we have a collagen fiber here, another collagen, sorry, a collagen molecule here, another collagen molecule, another collagen molecule, another collagen molecule. The bands show you the gaps between the collagen molecules. And many collagen molecules form a fibril. Then many fibrils form a fiber. So this is still a scanning electron micrograph of human collagen fibers. So these are the fibers. You can see each fiber is made up of many fibrils lying side by side. And these fibers are very large that you can even see them with an ordinary light microscope. So that was it for this lesson. I wanted you to know about globular proteins. I wanted you to know about collagen and to know about hemoglobin. Hemoglobin as an example of a globular protein and collagen as an example of a fibrous protein. So you should be able to describe the structure of hemoglobin and the structure of collagen. So if you have loved this video, please give it a thumbs up, share, encourage your friends to also come and watch, and then subscribe to our channel. If you are interested in learning about cell membrane and transport, it is our next topic. So go and watch it. See you on the next one. Bye.